All right, let's talk about one of the most incredible, high-stakes missions happening inside your body, probably even as we speak.
So imagine you get a cut or you pick up some kind of infection. That's an emergency. A 911 call goes out inside your body. But have you ever stopped to wonder how the first responders, your immune cells, actually get from the superhighway of your bloodstream to the exact street address of the problem?
Well, the answer is a process with an amazing name: diapedesis. You can think of it as the microscopic great escape that allows your white blood cells, those leukocytes, to literally leave the circulatory system and become the heroes on the ground where they're needed most.
So how does it all start? Well, just like any great rescue mission, it begins with a desperate call for help. When your tissue gets damaged, it doesn't just sit there. It releases a flood of chemical signals called chemokines. These are, for all intents and purposes, a biological distress beacon. It's that cellular 911 call broadcasting the exact location of the emergency to any immune cells that might be passing by.
And you bet that call gets heard. It's heard by our first responders, the leukocytes, which are constantly on patrol, coursing through your bloodstream.
But here's the thing. This isn't some chaotic mad dash. No, it's a very precise 4-step mission plan. First, the cell has to slow down and start rolling. Then it gets activated by those chemical signals. Next, it has to lock on tight to the vessel wall. That's adhesion. And only then can it execute the final escape, transmigration.
Let's take a look at that first step, because it's pretty cool. The blood is flowing fast, right? It's like a freeway. A leukocyte can't just slam on the brakes. So instead, it starts to kind of tumble and roll along the inner wall of the blood vessel, making these quick, fleeting connections that slow it down so it can look for the right exit. So how does that even work on a molecular level? Well, the wall of the blood vessel puts out these molecules called selectins. You could think of them as tiny, sticky speed bumps. The leukocyte has its own molecules that briefly grab onto these selectins. And that's what causes that characteristic rolling motion. It's really just a series of perfectly timed molecular handshakes.
Okay, so our leukocyte has slowed its roll, it's near the emergency site, and now it's time to gear up and get ready to stop completely.
This is where those chemokines, that 911 call we talked about, become absolutely critical. As the leukocyte rolls past the source of those signals, they latch onto its surface and act like a switch, flipping the cell from patrol mode into full-on action mode. This activation triggers a massive change in another set of molecules on the leukocyte surface, called integrins. They switch from what we call a low affinity, or weak grip state, to a high affinity, strong grip state. It's like the cell suddenly deploys a set of powerful grappling hooks.
Now, with its grappling hooks out, the leukocyte latches on hard to the corresponding molecules on the vessel wall. And this incredibly powerful bond is what finally brings the speeding cell to a dead stop, right at the exit ramp for the emergency. And that is the crucial transition. The cell goes from making these weak, temporary handshakes for rolling to forming these strong, unbreakable bonds for adhesion. It's gone from a slow cruise to being completely locked on, ready for the final and most dramatic step of all.
And now for the main event. The leukocyte is locked in place. It now has to perform its incredible escape act, breaching the wall of the blood vessel itself.
And here is the part that just blows my mind. The cell doesn't smash through the wall. It finds the seams. It literally starts to change its own shape, to contort itself so it can squeeze through the tiny gaps between the cells that make up that wall.
And it's not just fumbling around blindly. This is made possible by a specific molecule called PCAM1 that acts like a guide, or maybe like the teeth on a zipper. It's located right at the junctions between the wall cells, and the leukocyte uses it to find the weak point and help pull itself through that impossibly tight space. It is a genuine feat of cellular acrobatics.
And just like that, it's through. The escape is complete. The leukocyte is now officially out of the bloodstream and inside the damaged tissue. And from there, it just follows that breadcrumb trail of chemokines directly to the invaders or the injury site to do its job. Mission successful.
Now, this process is absolutely 100% essential for our survival. But what happens when this perfectly choreographed mission goes wrong? Well, it turns out our hero can sometimes play the part of the villain. The reality is that diapedesis really has two faces. On one hand, it's the hero, right? The rapid response that fights infection, cleans up debris, helps repair tissue. But when the system gets dysregulated, when too many cells get called in, or they stay for too long, or they show up at the wrong address. That exact same process becomes a source of chronic, damaging inflammation. And that is at the heart of so many diseases. And this isn't just a theory. This process going rogue is a key player in some truly devastating conditions. In rheumatoid arthritis, these first responders mistakenly invade and destroy the lining of our own joints. After a heart attack or a stroke, a flood of these cells rushing into the damaged area can actually make the injury worse. And, maybe most sinister of all, some cancer cells actually learn to hijack this very escape mechanism to spread throughout the body.
But ultimately, diapedesis is one of biology's most elegant solutions. It's a microscopic ballet of molecules and cells, a rapid deployment system that is constantly on guard, protecting you from countless threats every single day of your life. And that kind of leaves us with a final thought, doesn't it? We rely on this amazing system to save us, but the whole thing hinges on the accuracy of that initial 911 call. So what happens when the system gets confused? When the first responders can no longer tell the difference between an actual enemy and the very body they are sworn to protect? That's a question scientists are working tirelessly to answer.